Hello viewers. In my today's lecture, I will discuss on the importance of non-essential and essential amino acids, their biosynthetic pathways, and the two important methyl group donors, S-Adenosyl Methionine and tetrahydrofolate. First of all, I will discuss on what are essential amino acids. There are many amino acids whose biosynthetic pathways are present only in plants and microorganisms. But humans must have these amino acids in their diet. So these amino acids are known as essential or nutritionally indispensable amino acids. Examples include histidine, isoleucine, leucine, lysine, methionine, etc. Arginine is another amino acid which may be classified as essential, although it can be synthesized from urea cycle as it is required in greater amounts than it can be produced by that route for normal growth and development of children. The essential amino acids occur in animal and vegetable proteins, but different proteins, however, contain different proportions of these amino acids. So a balanced protein diet supplies the proper proportions of all of these essential amino acids. Now, what are non-essential amino acids? Man can make 11 out of these 20 amino acids from very common intermediates. So these amino acids are called non-essential or nutritionally dispensable amino acids. Examples include alanine, arginine, asparagine, aspartate, cysteine, glutamate, etc. Now, giving you an idea of what are non-essential and essential amino acids, I will now switch over to amino acid biosynthesis. Amino acids play a very important role in the metabolism of all organisms. Amino acids are precursors of proteins. All amino acids excepting glycine are chiral, so the stereochemical control is very much important in this biosynthesis, and the correct isomer must be produced with high fidelity in the biosynthetic pathways. First, I will discuss on the biosynthesis of non-essential amino acids. As I have already told that these non-essential amino acids can be synthesized from mainly 4 common metabolic intermediates like pyruvate, oxaloacetate, alpha-ketoglutarate and 3-phosphoglycerate. These non-essential amino acids have simple biosynthetic pathways excepting tyrosine, but tyrosine can also be synthesized from one-step hydroxylation from the amino acid phenylalanine. First of all, I will discuss on the biosynthesis of the non-essential amino acid glutamate. Glutamate can be synthesized from glutamate synthase in microorganisms, plants and lower animals. Now, the reaction between NADPH H+, glutamine and alpha-ketoglutarate results in the production of glutamate and NADP+. Here in this particular slide, we can see that how the different subunits like the alpha subunit and the beta subunit helps in the generation of glutamate. Glutamate can also be synthesized by another enzyme called glutamate dehydrogenase. The reaction between ammonium ion, alpha-ketoglutarate, NADPH and H+ results in the generation of glutamate, NADP+ and water. There is an alternative pathway of glutamate biosynthesis in some of the organisms. There, the reaction between ATP, glutamine and aspartate results in the generation of glutamate, asparagine, AMP and pyrophosphate. Another important non-essential amino acid is asparagine, and in this particular slide we can see how the reaction between aspartate and glutamine results in the production of asparagine and glutamate, The enzyme catalyzing the reaction is asparagine synthetase, and this reaction requires ATP. Asparagine biosynthesis in bacteria takes place following a different route. There, the reaction between aspartate, ammonium ion and ATP results in the production of asparagine, AMP, pyrophosphate and free protons. Glutamine is even another non-essential amino acid that can be synthesized by glutamine synthetase from glutamate in mammals. Here in this particular slide we can see the synthesis of glutamine takes place via an intermediate known as gamma glutamyl phosphate. This synthesis pathway also requires ATP. Glutamine synthetase is a very important enzyme. Now glutamine serves as amino group donor. So the pivotal position of glutamine synthetase makes its control a vital aspect of this process. Now I will discuss on the regulation of mammalian as well as bacterial glutamine synthetase. Now mammalian glutamine synthetases can be activated by alpha ketoglutarate, which is produced from oxidative deamination of glutamate. This type of control prevents the accumulation of ammonia, that is the product of this reaction. Now coming into the regulation of bacterial glutamine synthetase, as we can see in this slide that it is 12 identical 469 residue subunits, and it can be controlled by various types of effectors. For example, in bacteria Salmonella typhimurium, glutamine synthetase can be complexed with glutamate structural analog, that is phosphinothreonine. In this slide, the structure of phosphinothreonine is shown. Also, 9 feedback inhibitors can regulate bacterial glutamine synthetase. These 9 feedback inhibitors are shown in this slide. These are tryptophan, histidine, glucosamine 6-phosphate, carbamoyl phosphate, CTP, AMP, alanine, glycine and others. Bacterial glutamine synthetase in E. coli can also be regulated by covalent modification of a specific tyrosine residue by the enzyme adenylyl transferase, and the reaction is adenylylation. Now after discussing the biosynthesis of the non-essential amino acids like glutamate, asparagine and glutamine, I will now discuss on the biosynthesis of glutamate family of amino acids. First of all, in this slide we can see the biosynthesis of proline. Glutamate family of amino acids means all of these amino acids are synthesized starting from L-glutamic acid. Now here in this slide we can see that L-proline is synthesized following a multi-step pathway starting from L-glutamic acid. 4-hydroxyproline is another important amino acid which can be produced from proline through hydroxylation reaction. Arginine is another important amino acid which also falls under glutamate family of amino acids, and this amino acid can be synthesized, as we can see in the slide, through ornithine cycle. This I have discussed in my previous lecture that ornithine cycle consists of 5 steps, and arginine is generated in the 4th step from argininosuccinic acid releasing fumarate and arginine. The enzyme catalyzing the reaction is argininosuccinate lyase. Ornithine is another important amino acid also synthesized from the starting compound L-glutamate. Ornithine can also be synthesized by an alternative pathway. The enzyme catalyzing the reaction is ornithine delta aminotransferase. Now serine is another amino acid which is produced from 3-phosphoglycerate. Alternative pathway of serine biosynthesis also exists in some organisms, where hydroxypyruvate generated from D-glycerate undergoes a transamination reaction with either alanine or glycine, thereby resulting in the generation of serine and either pyruvate or glyoxylate respectively. Now here in this particular slide we can see glycine biosynthesis which follows 2 pathways. In one of the pathways, glycine can be produced from serine. The enzyme catalyzing the reaction is serine hydroxymethyl transferase, and another route is from carbon dioxide and ammonium ion, and the enzyme is glycine synthase. Now, cysteine biosynthesis takes place in animals from homocysteine and serine. Now, there are two genetic defects, namely homocysteinuria and cystathione. In the first condition, there occurs excretion of homocysteine in urine, and that is known as homocysteinuria. Now, homocysteine accumulation cause the patients to have mental deficiencies, and they also have the tendency of coronary heart disease and arteriosclerosis. Homocysteine accumulation also raise oxidative stress. Another condition is cystathione. Cysteine biosynthesis in plants and microorganisms takes place following two routes. One is from serine and another is from ATP and sulfate ion. Now, after discussing the importance of non-essential amino acids and their biosynthesis, I will now discuss the biosynthesis of essential amino acids. As I have already told that these essential amino acids are present only in plants and microorganisms, but humans must have these amino acids in their diet. These amino acids have complex biosynthetic pathways, and they are unlike non-essential amino acids which have fewer than 5 steps in their biosynthesis. Now, first of all, we will have a look into the aspartate family of essential amino acids, which means that all these groups of amino acids are generated starting from aspartate. These groups of amino acids are threonine, methionine and lysine. In this particular slide, we can see the synthesis of these 3 amino acids starting from aspartate. Now, alternative pathway of threonine biosynthesis also occur, where the reaction between acetaldehyde and glycine results in the production of L-threonine. The enzyme catalyzing the reaction is serine hydroxymethyl transferase, and the coenzyme is pyridoxal phosphate or PLP. Now, there are 2 direct methyl group donors in case of methionine biosynthesis. These are betaine and dimethylthetine. This particular slide shows the structure of these 2 methyl group donors. Now, lysine biosynthesis is another important biosynthetic pathway, and in this particular slide we can see that there are 2 major pathways. In bacteria and higher plants, lysine biosynthesis follows the diaminopimelic acid pathway, whereas in most of fungi, lysine biosynthesis follows alpha amino adipic acid pathway. Most of the metabolic reactions are controlled in the first committed step, and in this case also the group of amino acids, the threonine, methionine and lysine, are controlled in the first step, and the enzyme catalyzing the first step is aspartokinase. There are 3 isozymic forms of aspartokinase, which is aspartokinase 1, aspartokinase 2 and aspartokinase 3. In the following chart, we can see how these 3 isozymes of aspartokinase are controlled by these amino acids. In case of aspartokinase 1, the feedback inhibitor is threonine and the co-repressors are threonine and isoleucine. In case of aspartokinase 2, the co-repressor is methionine, and in case of aspartokinase 3, both the feedback inhibitor and co-repressor is lysine. Coming into the pyruvate family of amino acids, first of all, we can see in this slide the biosynthesis of valine, and it falls under pyruvate family, means that it is synthesized from the starting compound pyruvate. Now, the biosynthesis of lysine, we can see in this slide that it also follows a complex multi-step pathway, and also the starting compound is pyruvate. Next, we can see in this slide the biosynthesis of isoleucine, which also follows a multi-step pathway, and the starting compound is pyruvate. Let us discuss the biosynthesis of another three important aromatic amino acids, which are phenylalanine, tyrosine and tryptophan. All these amino acids are synthesized from a common compound called chorismate. Now in this particular slide, we can see the biosynthesis of chorismate starting from phosphenol pyruvate and erythrose 4-phosphate. From chorismate, three amino acids are produced, which are tryptophan, tyrosine and phenylalanine. This slide shows the biosynthesis of tryptophan starting from chorismate and follows a complex multi-step pathway. Again, tyrosine and phenylalanine are also produced from chorismate following two different routes. But as I have already told that tyrosine may also be produced through one-step hydroxylation of the amino acid phenylalanine, and the enzyme catalyzing the reaction is phenylalanine hydroxylase. But many microorganisms synthesize tyrosine directly from prephenate that I have discussed earlier. One important information is that glyphosate, the commercial name of which is Roundup, is the most active ingredient in majority of the weed killers, and it is a broad spectrum herbicide. Now this herbicide inhibits the enzyme that generates the compound 5-enol pyruvyl shikimate 3-phosphate and hence blocks aromatic amino acid biosynthesis. In this particular slide, we can see the structure of glyphosate. Now because animals lack this enzyme, this herbicide is non-toxic to animals. Now in this particular slide, we can see the mechanism of action of glyphosate. As I have already told that it inhibits the formation of 5-enol pyruvyl shikimic acid 3-phosphate and thereby inhibits the formation of chorismic acid. And as I have said earlier, that chorismic acid is the precursor compound for the three amino acids phenylalanine, tyrosine and tryptophan. So chorismic acid formation is inhibited, and as a result there occurs a blockage of the synthesis of these three amino acids. Next, we can see in this slide histidine biosynthesis, which is another important essential amino acid. The starting compounds are 5-phosphoribosyl 1-pyrophosphate and ATP. As the slide shows, there are mainly three major control points. The first one is called feedback control. As the diagram shows, that in the conversion of L-threonine to L-isoleucine, it follows 5 major steps. Now the end product, what it does, it inhibits the first enzyme of the pathway, and here it is L-threonine dehydratase, and hence it controls its own amount. This is known as feedback inhibition. So this is one type of regulation. Another way of regulation is by allosteric control, and the third way of regulation is through repression of the necessary biosynthetic enzymes. One of the interesting information is that we know that amino acids are precursors of proteins, but we must also know that amino acids serve as precursors for many other small molecules which have important biological roles. The most important of these are adenine, cytosine, sphingosine, histamine, thyroxine, epinephrine, serotonin and nicotinamide unit of NAD+. The importance of two methyl group donors, which are S-Adenosyl Methionine and tetrahydrofolate. These are very important methyl group donors and are required in most of the biosynthetic pathways. First of all, I will discuss about S-Adenosyl Methionine or SAM. Now the advantage of S-Adenosyl Methionine is that it has high group transfer potential than tetrahydrofolate, so it is utilized in most of the biosynthetic pathways. This particular slide shows the structure of S-Adenosyl Methionine. This slide shows the biosynthesis of S-Adenosyl Methionine. It is produced from methionine and ATP with the generation of S-Adenosyl Methionine and release of pyrophosphate and inorganic phosphate. This slide shows activated methyl cycle. Let me explain it to you. S-Adenosyl Methionine is a potent alkylating agent, all because of the presence of its destabilizing sulfonium ion. Now in this cycle we can see that S-Adenosyl Methionine, after donating its methyl group, generates a methylated acceptor and itself gets converted to S-Adenosyl homocysteine. It then produces homocysteine with the release of adenosine. From homocysteine, methionine is generated with the help of N5-methyl tetrahydrofolate. From methionine, again S-Adenosyl Methionine is regenerated with the reaction of ATP. So this is the activated methyl cycle. Advantage of S-Adenosyl Methionine or SAM, as I have already told, that it is 10 to the power 3 times more efficient than tetrahydrofolate, as its group transfer potential is very much high. But the disadvantage is that it carries the 1 carbon unit only in one oxidation state, that is -3. Whereas tetrahydrofolate carries 1 carbon unit in 3 oxidation states, which are -2, 0 and +2. Lastly, I will discuss the importance of another methyl group donor, which is tetrahydrofolate. This particular slide shows the structure of tetrahydrofolate, which is a bit complex. It consists of 3 major parts, which are 2-amino-4-oxo-6-methylpterine, para-amino-benzoic acid and glutamic acid residues. Now the first 2 taken together are called pteroic acid, and the 3 units taken together is called pteroyl glutamic acid or tetrahydrofolate. Now the source of tetrahydrofolate is folic acid, which is vitamin H, as we all know, which is the doubly oxidized form of tetrahydrofolate. Now the source of folic acid is from diet or normal intestinal microflora. Tetrahydrofolate is produced, as we can see in this slide, through 2-step reduction from folate and dihydrofolate. First, folate is reduced to dihydrofolate and dihydrofolate is then reduced to tetrahydrofolate. The enzyme catalyzing this 2-step reduction is dihydrofolate reductase. Let us discuss the role of THF in metabolism. It transfers 1 carbon groups like methyl, methylene, formyl, formimino, methinyl, etc. This 1 carbon unit can be bonded to either N5, N10 or both N5 and N10 of tetrahydrofolate. Now this particular chart shows how the different 1 carbon group are bonded to N5 or N10 or both N5 and N10 and results in the generation of various derivatives of tetrahydrofolate. And as I have already said, that tetrahydrofolate can carry this 1 carbon group in 3 oxidation states, which are -2, 0 and +2. This slide shows the various derivatives of tetrahydrofolate, their structures, and these derivatives of tetrahydrofolate can be interconverted amongst each other. Advantage of THF, that it can transfer the 1 carbon group in 3 oxidation states, -2, 0 and +2, but the disadvantage is that its group transfer potential is much less than S-Adenosyl Methionine. So S-Adenosyl Methionine is the preferred methyl group donor, unlike THF. in my today's lecture, we have studied the importance of essential and non-essential amino acids, their biosynthetic pathways, and also the importance of the two methyl group donors, tetrahydrofolate and S-Adenosyl Methionine. Thank you. This is all for today's lecture.